Hello, everyone, and welcome to our Tea Time series webinar. So today we would like to introduce Reni Kitte, MSc from the Institute of Fraunhofer Institute of Immunology and Cell Therapy. Reni was so kind to join us today. She will be discussing the transient expression of mRNA-LNP-based Chimeric antigen receptor for safe immunology therapy. Now, before we start, I would like to introduce you to our team. So myself, I'm Suha, and I am based in London, UK, and I work closely with our colleague, Jorgen, who is based in Germany, and of course, our P&I gurus, who are the experts in our technology. So feel free to reach out to Martin in Germany, Edward in the UK, and Sarah in France. Reni, over to you.
Yeah, hello everyone. Welcome also from my side to today's Tea Time webinar. So yeah, before I start, I would like to thank Precision Nanosystems, especially Martin, for inviting me for today's webinar series and for giving me the opportunity to present my data in this webinar series. I'm really happy about it. And yeah, today's topic is the transient expression of chimeric antigen receptors, in short CARs, via mRNA lipid nanoparticle-based transfection for safe immunotherapy. And before So I would like to start my presentation with a short introduction about CAR T-cell therapy before I come to its limitations and challenges. So basically, CAR T-cell therapy starts with the collection of the white blood cells from the patient. So the patient undergoes leukapheresis, and after shipping it to the manufacturing site, the T cells are isolated and then genetically modified by a viral transduction to express the chimeric antigen receptor on the T cell surface. These CAR T cells are then expanded, and few weeks after leukapheresis, the CAR T cells are reinfused back into the patient. So that these CAR T cells via the CAR can specifically recognize and eliminate the tumor cells. But this manufacturing process has some limitations and challenges. And for instance, the poor patient T cell quality. This is because the patients underwent a lot of treatment cycles of chemo or radiotherapy. So they are of poor quality, which can inhibit the CAR T cell production. And and the next one is there are safety concerns such as the risk for insertional mutagenesis. This is because the virus stably integrated the DNA, so the CAR encoding DNA, into the T cell genome. So it provides permanent modification, but this is associated with the safety concerns. So the integration is on a random location. And along with this permanent modification, this stably transduced CAR T cells can cause long-term side effects such as cytokine release syndrome, neurotoxicities, or the depletion of healthy cells. And one approach to address these challenges would be the use of transient CAR T cells. And in this case, we are also working with the patient T cells and isolated them, but the cells are expanded first before it comes to the modification to CAR T cells. And this is not performed by viral transduction, but by the transfer of the CAR encoding mRNA. And this approach would circumvent these challenges, so the poor patient T cell quality, because the T cells are expanded first so they can recover during in vitro culture. mRNA does not integrate into the genome, so there is no risk for insertional mutagenesis. It is a non-permanent modification, and thus also no long-term side effects are expected. Another advantage is that this approach has a high potential for an in vivo application, meaning that in combination with an appropriate delivery system, such as the lipid nanoparticles in which the mRNA is packed, are then applied to the patient to directly produce the CAR T cells in a non-permanent way directly inside the patient. So the mRNA lipid nanoparticles are applied to the patient, and this would provide an off-the-shelf product since it's not patient individual anymore, and this would also be fast and cost-effective to produce. And another issue when thinking about CAR T cell therapy is in general is that there are no tumor-specific targets, only tumor-associated targets, meaning that also healthy cells can express this antigen, which it's targeted by the CAR T cell, and the CAR T cell is not able to discriminate between healthy and the tumor cells. So it's even more important to provide a non-permanent modification in order not to deplete the healthy cells for a long time. So I started with the production of the mRNA by in vitro transcription using T7 polymerase, and it starts with a linear DNA template. This can be performed via a linearized plasmid DNA when the T7 promoter sequence is already present, but it's also possible via an PCR product, as in my case. So I simply added the T7 promoter sequence, since it's not present, via the forward primer. So from this template, I performed in vitro transcription to generate RNA and RNA processing to add the 5' cap structure and the 3' poly A tail. And what I've done first is I analyzed a non-processed RNA, which does not have the cap structure and the poly A tail, as well as partly processed RNA, which has either the poly A tail or the cap structure, and fully processed RNA, which have both structures. And in the gel, you can nicely see the size or the length differences between polyadenylated and non-polyadenylated mRNA. And I analyzed these mRNAs in HEC293 cells. Therefore, I packed the mRNA into liposomes to transfect the HEC cells, which then expressed the CAR on their surface, and the CAR expression was then analyzed via flow cytometry using an anti-Fab2 antibody, which is conjugated to enfluorochrome elexaflor 647 in this case. And this antibody recognizes antibody structures, which are also part of the CAR. So it's a really nice and universal CAR detection method. And when looking at the results, so the CAR expression, the proportion of CAR positive cells, as well as the translation efficiency of the CAR positive cells indicated by the median fluorescence intensity, it's nicely shown that only the fully processed RNA, so having both the cap and the poly A tail, led to comparable results to the reference, which was plasmid DNA in this case. So also plasmid DNA was transiently transfected. So yeah, it is already known, but we confirmed it that RNA processing is essential for sufficient translation. We then optimized the mRNA for a high stability, low immunogenicity, and high translation efficiency. Therefore, we analyzed different nucleotide modifications. So, for example, the replacement of uridine by N1-methyl pseudouridine, but we also worked with untranslated regions and also the removal of double-stranded RNA, since this is known to induce unwanted immune responses. And in general, when there are no modification inside the mRNA. As I shown in my slide before, the CAR is expressed. But we then observed a high immunogenicity and low stability. But in contrast, when adding many modifications, so several different modified nucleotides, for instance, then we saw a high stability and low immunogenicity, but then the translation efficiency was low. So when optimizing the mRNA for your approach, you should keep in mind that there is a sensitive balance between stability, immunogenicity, and translation efficiency of the mRNA, and that this effect is also RNA dependent and cell type dependent. So after optimizing the mRNA, we established different mRNA delivery methods, and the current state of the art used in clinical trials which investigates transient CAR T-cell therapy is electroporation. So we established electroporation and as an alternative also lipid nanoparticles. And therefore we use the Genvoy ILM T-cell kit for mRNA from Precision Nanosystems. This is a kit which is optimized for T cells, and this comes with a very easy workflow. So it starts on day 0 with the isolation of peripheral blood mononuclear cells, PBMCs, from Buffy code, and from that the isolation of the T cells and their activation. There are also kits available where you can directly isolate the T cells from the Buffy code, but yeah, in our lab we usually use the way via PBMCs. On day 3, after isolation and activation, the mRNA lipid nanoparticles are formulated, and this is also very simple and easy. Therefore, we use the nano assembler Spark device from Precision Nanosystem together with its specialized cartridges. So in this cartridge, there are 3 cavities. In the biggest one, you simply add a buffer. In the second one, the mRNA dilution. And in the third one, the lipid mix. Then you simply cap the cartridge, insert it into the Spark device, push the one and only button, and within very few seconds, you can collect your formulated mRNA lipid nanoparticles from the biggest well, and they are ready for transfection of T cells. And for our first experiment with this kit, we used an mRNA which encodes for enhanced green fluorescent protein, so EGFP, as a model mRNA. And when adding no lipid nanoparticles, of course, we did not see any EGFP expression, and the viability of the T cells was 90%, so it serves as a reference here. And when adding 1 microgram per milliliter mRNA, the EGFP expression was really high, so over 90% were EGFP positive, while the viability was not affected. And when adding a little bit more, so 4 microgram per milliliter, the EGFP expression was slightly increased, although it was already high, but the viability decreases a little bit, but this in a very acceptable range. So for the generation of transient CAR T cells, we adapted the workflow a little bit. So we did not transfect the T cells on day 3, but on day 7 in order to expand the T cells due to 2 reasons. One is our used CAR detection method. As I already mentioned, therefore we used an anti-Fab2 antibody, which recognizes antibody structures such as the CAR, but also antibodies which are bound on the activated T cells, since for instance, an anti-CD28 antibody, which is used for activation of T cells. So on day 3, when we transfected on day 3, we have a really high background since due to this activation antibodies, but on day 7, it's no issue anymore. But most important, we let the T cells expand due to the ex vivo transient CAR T cell production process, where we need to expand the T cells in order to get a high cell number. But before we transfected the HEC cells, we analyzed the mRNA lipid nanoparticles in more detail. We analyzed the mRNA recovery and encapsulation efficiency. Therefore, we used the QuantIT ribocrene RNA reagent and kit from Invitrogen. And this assay is based on breaking up the lipid nanoparticles by Triton X to release the mRNA, which then allows the quantification by binding to the ribocrene fluorescent ribocrene reagent. And we were able to recover 70 to 80% of the mRNA, and the encapsulation efficiency was nearly 100%. We also analyzed the size of the LNPs, which was about 70 and 80 nanometers in diameter. We also analyzed empty LNPs. They were a little bit smaller. We also analyzed the polydispersity index. This should be as low as possible, so the results look quite good. And for this analysis, we used the Cedars-Heiser, which is a dynamic light scattering device for particle analysis. We performed a head-to-head comparison between electroporation and lipid nanoparticle-based transfection. And for electroporation, we used our established protocols using the neon transfection system from Thermo Fisher. And of course, the GeneVoy ILM T-cell kit for RNA from Precision Nanosystems. And again, the workflow, so on day 0, the isolation of the PBMCs, therefrom the isolation of the T-cell and activation. On day 7, the transfection of the T-cells, either by electroporation or by mRNA-LNP-based transfection. And the analysis already starts on the same day, so already after 6 hours after transfection, and then in a daily manner. Regarding the viability, so the differences were most pronounced during the early time points. So after 6 hours, 1 day, and 2 days post-transfection. And in gray, you see the untransfected T cells as a reference for each day. You see in lighter colors the water transfected T cells, in blue the electroporated T cells, and in red the T cells which received empty LNPs. And in the darker colors, you see the T cells which received the CAR mRNA. And you can already see it on the water transfected T cells but it was most pronounced when they received the mRNA, that electroporation is more harmful to T cells than the LNP treatment, because the viability drops down to 30% after electroporation, while it was about 70% after LNP treatment. Regarding the CAR expression, so after electroporation, 6 hours post-transfection, you already have over 90% of CAR expressing T cells already after 24 hours, but then it rapidly decreases until it was nearly not detectable after 3 days post-transfection. In contrast, after LNP treatment, the peak CAR expression was after 24 hours. Then it also decreases, but much more slowly. So with LNP treatment, you received a prolonged CAR expression by about 2 days. And when also looking at the CAR expression level, so the median fluorescence intensity, you can nicely see that already after 6 hours after electroporation, the peak CAR expression was reached and then decreases. So after electroporation, what I already said, the peak CAR expression was after 6 hours compared to the LNP-based transfection, where the peak CAR expression was after 24 hours. And these differences can be explained by the different mechanisms the mRNA enters the cells. So for electroporation, the mRNA is naked, and once it enters the cells, it's directly available for translation. While for LNP-based transfection, the LNPs have first to be taken up by endocytosis and then released from the lipid nanoparticles and the endosome, and this takes some time. But consistent to the CAR expression, also the efficacy of the transfected CAR T cells was prolonged after when the CAR T cells derived from the mRNA lipid nanoparticles. And again, the peak efficacy of electroporated CAR T cells is already after 6 hours, while it was after 24 hours for the LNPs. So we see a prolonged killing by the mRNA lipid nanoparticles generated transient CAR T cells. And when looking at the state of the art, which is electroporation with this lipid nanoparticles, we could prolong the therapeutic window of at least 2 days when using the mRNA lipid nanoparticles. And last, we then compared the mRNA lipid nanoparticle-based transfection or based CAR T cells with CAR T cells which are stable, so generated via viral transduction. So we received a high CAR expression with this transduction, but also with a transfection. So we see comparable CAR expressions for 2 days post-transfection compared to the viral transduced CAR T cells. And on day 2 post-transfection, also the efficacy against a tumor antigen positive cell line, KG1 in this case, was comparable between stable and the transient CAR T cells. And what I would like to show here, against the tumor antigen negative cell line, so K562 cells in this case, you can see no effect, so that this activity of these cells is really CAR dependent. So 2 days after transfection, the transient CAR expression and the killing efficacy were comparable to the stable CAR T cells. And with that, I would like to summarize my talk a little bit. So we established the mRNA delivery for 2 methods, the electroporation and lipid nanoparticle-based transfection. And we were able to show that mRNA lipid nanoparticles outperform electroporation regarding viability and duration of CAR expression and killing efficacy. And when comparing these transient CAR T cells with the stable CAR T cells, they were also as efficient as the stable CAR T cells in our in vitro assays. And with that, I would like to thank my colleagues from the Fraunhofer IZI, especially the ATMP engineering unit, and especially Dr. Sandy Tretbar for her excellent supervision, as well as Precision Nanosystems as our collaboration partner, especially Martin and Jürgen and our other collaboration partners. And yeah, also you for your attention. Thank you very much.
Thanks a lot for the talk, for presenting your amazing data, for your time. To the audience, thanks for being here today with us, asking all these questions, staying with us for the whole discussion, and I wish everyone a very good end of the summer. And see you soon. Bye-bye.
Bye-bye.